All right, everybody. So, sex-linked traits and pedigrees. So, we have done genetically linked traits, but now we're getting into sex-linked. And we're going to do a little bit of backtracking before we start getting into what's actually going on there. So again, if we're talking humans, we have 23 pairs of homologous chromosomes, and 22 of those are autosomes, which do not determine gender, but we do have sex chromosomes. And I quickly want to go over differentiating between sex chromosomes and sex cells because sometimes that gets people confused. Sex cells are gametes. Gametes are sperm and egg. Now, in different organisms, they might not have sperm and egg, they might have pollen and different things like that, but they still have haploid versions of certain things that will pass on to the next generation when fertilization happens. So sex cells, if I were to have a karyotype of a sex cell, like an egg or a sperm, I wouldn't have um, 46 chromosomes here. I'd only have 23, and I'd only have one X or one Y, and that's very different than this. So sometimes when people hear sex chromosome, they start thinking sex cells, when in reality, you've got trillions of cells in your body, and every single one of those cells contains sex chromosomes. So if you are a boy, if you're male, every single cell in your body is going to have X and Y. And there are other genes on these that do other things for your body besides determine your gender, which we're going to get into when we start talking about color blindness. And if you're a female, every single cell in your body is going to have X and X besides sperm and egg. So sex cells, gametes, haploid, meiosis, only half of the total number of chromosomes, but when we're talking sex chromosomes, we're talking about basically the 23rd pair of chromosomes. So, moving on here, we spend a lot of time talking about Punnett squares, Punnett squares, Punnett squares. And we're going to talk about Punnett squares again today. And the thing is, I just want to mention that Mendelian genetics is typically overemphasized because it is rare that a trait is determined by a single gene. So even though we are going to do this, most of the time, this is not really that applicable. It does get in when we start talking about diseases because a lot of diseases are because of proteins that are the wrong shape, which can come from mutation and then get passed on a recessive version of that. But a lot of modern genetic research isn't just about like what genes exist inside an organism, because what we find is as we keep on sequencing genomes, species that seem very unrelated can actually have the same genes. They have the same proteins within many, many, many different living things. So modern genetic research a lot of times focuses on turning them on or turning them off and the expression of common genes instead of different genes. Okay, so moving in, the first thing I want to talk about and reemphasize is that when we are coming up with this dominant and recessive and big letters and little letters and genotypes and phenotypes, everything does come back to proteins. So I want to talk about the hemoglobin protein for a second. So red blood cells make a tremendous amount of hemoglobin protein prior to ejecting its nucleus and giving it its concave shape. And scientists actually can track down exactly where the code for hemoglobin protein is, and they find that it's on our 11th autosome. So we can track down and we can actually find the exact same location, the exact same nucleotide sequence to determine how the protein is made. What letters make code for which amino acids in which order ends up making the hemoglobin. So then when they find out, okay, sickle cell anemia is a mutated form of hemoglobin, they can actually track this misshapen protein all the way back to which letters of the code are incorrect on the particular chromosome. So when we start talking about alleles and traits and everything like that, we got to understand we're coming back to the code, and genes code for proteins. Proteins determine traits. And slight variations in protein structure can ultimately result in different versions of alleles. So we can have disease because of a difference in the protein structure. And we're going to take a look at that. So first of all, mutated DNA often can lead to a broken protein. And typically we think of that as a bad thing. If your proteins are meant to be a particular shape, and if they're not that shape, they can't do the function that was meant for that protein. But is that always bad? And we're looking at this sickle cell anemia and this misshapen hemoglobin protein. And the thing is, it didn't start as a bad thing. So malaria is caused by this little guy right here. It's a eukaryote. It's called Plasmodium. And when you are infected by this, again, the mosquito is just the vector. It's what's spreading this guy around. So this guy gets into your blood cells, your red blood cells, and releases an enzyme that destroys hemoglobin. So when this starts infecting people and it gets put into someone's blood, and this blood is perfectly normal, it's actually really, really bad because this ends up destroying all of the hemoglobin protein and people die from malaria. So along comes a mutation that ends up making the hemoglobin a different shape, and it makes it sticky basically, which causes some of the red blood cells to be misshapen. So this initial mutated DNA makes a broken, basically, version of hemoglobin, but that's not a bad thing. That's actually a really good thing, because instead of dying of malaria really quickly because this thing is destroying all of the normal hemoglobin, these little critters can't get in and destroy this new version of hemoglobin. So these people don't have the side effects of sickle cell anemia, but they are malaria resistant. So all of these people that have normal hemoglobin are dying and not passing on their traits. When along comes a mutated, broken version, and these people actually are able to survive longer, and you actually have a version of this because this little guy is going to make a sickle-shaped, which would result in a sickle-shaped hemoglobin, and this one is a normal hemoglobin, but together they're going to actually improve fitness of an individual and allow them to live longer and pass on their genes to their offspring. Now, when they start passing on their genes, if you have two carriers for this particular type, you end up with the full-on sickle cell anemia, which is bad. Um, and half the people that have sickle cell anemia die before they turn 50. So it's not necessarily that they're culled from the gene pool. Um, However, they're going to have a lot of pain associated with this. Back in the day, if you're getting chased by a saber-toothed tiger, you're not going to be able to process and carry oxygen nearly as well. And if you are in oxygen debt for many, many, many years, that's going to cause a lot of organ damage over time. So when we get into this, we are really talking about two different traits: malaria resistance and sickle cell. So what trait are we talking about when we are defining dominant? Because when we define dominant, dominant is what phenotype shows up in a heterozygous individual. So this heterozygous genotype, when we're talking about the sickle cell trait, this person does not exhibit all of the symptoms that are associated with sickle cell. So we would say that the sickle cell trait is recessive and not displayed with this. However, if we are talking about the malaria resistance and we're representing that trait with an R, these people are malaria resistant and they are actually are dominant for malaria resistance traits. And this group right here is actually, if we were going to define this trait, these people that are normal for hemoglobin are actually homozygous recessive for malaria resistance. So again, this isn't so easy and so cut and dry. And it's not like one thing is dominating the other. Dominant is what shows up in the phenotype of heterozygous genotypes. So again, we don't often think about this malaria resistance and how a mutation can actually be good, but we are going to focus on the sickle cell. So again, most of it's going to follow in this direction. So like I was just saying, on the 11th autosome, you can find the gene for hemoglobin protein, and you can do that for many other things. For example, vision, and we're going to get into color blindness here, but just imagine how many different proteins and how Punnett squares are pretty difficult to do for someone with certain vision types. Because the whole process of being able to see something is determined by a tremendous amount of different proteins working together here. So when light comes through, it hits your retina, and with your retina, you have rods and cones, and you have this big optic nerve here, and the light stimulates these rods and cones, which sends the messages back up to the optic nerve and ultimately into your brain. So we are going to look at color blindness, but again, there's a lot of different types of color blindness, and there's a lot of different proteins that are involved. And ultimately, yes, we will look at the sex-linked color blindness, but scientists have located other genes that have to do with color blindness. This gene right here, is on the 10th autosome. This gene right here is on the first autosome. This gene right here is on the second, and this gene right here is on the 8th. So you can pinpoint which proteins are possibly mutated, which are causing color blindness. And it can be very complicated. We are going to happen to look at, though, X or sex-linked red-green color blindness. So again, let's talk about this a little bit because I think it's important to understand that we are talking about proteins here. When you see something, I just talked about light coming through and hitting the retina, and the light being absorbed with these rods and cones. So I'm going to do a couple things here where we successively zoom in. So now we're looking at these rods and cones. And these rods and cones are specialized neurons. They're sensory receptors. They're photoreceptors. And within these, there are these little membrane discs. And if we zoom in on that, these membrane discs contain pigments. And within these pigments are proteins. So pigments can absorb light, and now it gets into cellular communication. So when light hits this protein changes shape, which causes this G protein action here where GTP replaces GDP, which causes a part of this G protein to slide along the membrane, which then causes this trigger of GTP becoming cyclic GMP, which closes a channel, and then it ultimately stops the communication going to your optic nerve. If we zoom in even further here, down to this little pigment here. So this pigment, which absorbs light, is actually run through this opsin protein. And when light hits it and it works properly, this protein actually straightens out. And then that triggers the cellular signal to be sent in. I'm going to quick run over here because I want you, this is just so cool. So again, we have these stacked membrane discs, which results in proteins embedded in a cell membrane, which causes a chain reaction of events. What else does this look like? Remember, within our chloroplasts, we have all of these thylakoid membranes stacked up with pigments embedded into that membrane, which then when light hits it, they absorb it and cause a chain reaction of events. So, I mean, some of these structures aren't all that different here. Anyways, that opsin protein, we can actually track that opsin protein, which is involved with a pigment which absorbs red-green wavelengths of light to the X chromosome. And it's this bottom part of the X chromosome that actually is the code to make this particular opsin protein. So again, we have all of this stuff. We can track down the genetic code for making this. And this particular one in our rods actually does the absorption of this wavelengths of light. And if you cannot, if this is mutated, we are not going to absorb this, and then therefore it won't straighten and won't send a cellular signal into your brain that says what color you're seeing. So, all right, we're time to get into Punnett squares and genotypes. So if we are looking at genotype and phenotype, getting down to the protein structure, the normal version of this we would represent with a capital dominant letter here. And it works properly. Along comes a mutation that makes an opsin protein that when light hits it, it does not straighten out and cannot cause a cellular signal to happen. So we are going to represent this mutated version of this opsin protein with the recessive allele. So again, recessive is what is not shown in heterozygous offspring. So in order for a woman to have normal vision, we could have big A, everything is all good here. The opsins are the right shape and they're working. If we are talking about homozygous recessive, for a woman to be colorblind, typically, there's all this stuff about X chromosome inactivation too, but that's for a whole nother day. So if you're going to be colorblind here, they need both of these versions to be broken, basically. And therefore, you have a version of this protein that's mutated, that's passed on from your parents, that causes no cellular signal to be sent to your brain for red and green. So if we are looking at this as a carrier, I don't know why this got all messed up. Anyways, a girl with normal vision would be a carrier if they have one version that's normal, like this, and one that is abnormal. So if you are a man, you have to have this version. You have to inherit from your mom the normal opsin protein. If you inherit from your mom a broken version of the opsin, you are going to be colorblind. Let's make sure this works here. These are the right. All right. So let's jump right into some Punnett squares and stuff here with this stuff. So, all right. If a woman is a carrier for red-green colorblindness and a man has normal vision, what are the projected phenotypes of their progeny? Holy cow. So, first of all, let's figure out genotypes here. A woman is a carrier. What would that look like? Okay. First of all, from a chromosome standpoint, a woman is XX and a man is XY. A woman being a carrier would have a genotype that is one normal and one mutated version or not proper opsin. That makes them a carrier. They are carrying along the recessive allele even though they have normal vision. They do not have the trait. They do not have color blindness. So what would the man's phenotype be? It would be that, because if it would be a small a, it would be colorblind. So the man has normal opsin proteins, has normal vision, does not have the trait for colorblindness. So from this, what does that look like from a Punnett square point of view? So this would be dad, this would be mom. So during production of gametes, dad could pass this on or this on. Mom could pass on this chromosome or this chromosome. So we would write this that way and that way. And mom would be written like that. So now in our Punnett squares, we are going to project the phenotypes. In this situation, this box is this and this to make this. This, you'd match this up and this up. We'd have a normal vision male, normal vision carrier. Yeah, that shouldn't be a carrier. This one down here would be the carrier. They would also have normal vision. But this last box here, you would have a colorblind male. Okay, so again, when we start talking X chromosomes, we have to match up the alleles with the X chromosomes because it's sex linked. It's linked on the X, not on the Y. So of 4 possible children, 2 would be male, 2 would be female, 1 would be normal, not a carrier, 1 would be normal and a carrier, 1 would be colorblind male, and 1 would have normal vision. Okay, next one. If a man is red-green colorblind, does it guarantee that his sons will be? So if we're looking at this, again, we need to figure out what our genotypes are going to be. And as far as genotypes go, when we are trying to think of whether or not this is guaranteed, we would think about, okay, man is red-green colorblind. This is how it would be. Does he pass it on to his sons? Does it guarantee that his sons will be this way? So let's put that there. Let's put that there. Let's figure out our gametes. Let's figure out the genotypes for our gametes. And then let's figure out our children. This one's going to be a female. That's a carrier. This one is going to be a male with normal vision. This one is going to be normal vision and a female that's also a carrier. And this one, again, is going to be normal vision. So If a man is red-green colorblind, does it guarantee his sons will be? No, it does not guarantee, because he is colorblind because of his X chromosome. His sons do not inherit his X chromosome, they only inherit his Y. So as long as mom is not a carrier, a man with red-green colorblindness will not have sons that are colorblind. It is not passed through the dad, it's passed through the mom. So, can a woman be red-green colorblind? Under what circumstances? What would it take to get a red-green colorblind woman? And this, we're going to stay away from X inactivation for now. But, think about what genotype would be required for a woman to be red-green colorblind and not just a carrier. And if you thought X small a, you would be right. How do we get to X small a, X small a? You would have to have mom be a carrier, and you'd have to have dad being colorblind. So again, mom would have to be a carrier, dad would have to be colorblind. And that would look like that, a carrier, and dad would be colorblind. Of course, if mom is colorblind, then it defeats this purpose. But how do we get a woman who's colorblind in the first place? We take dad and mom. Dad is colorblind. Mom is a carrier. Again, XAY, XAXA, carrier, colorblind, normal, colorblind female. That's where it comes from. From this X passed on from colorblind dad and this X passed on from carrier mom, we end up with this situation right here. Okay, I think that's long enough for this first one. That gets into all of sex-linked, but it really starts making sense once you start looking at pedigrees. But I think this video is long enough, so I'm gonna stop this one and start back up with pedigrees.